In the present symposium titled Progress on Personality Neuroscience Research and Individual Differences, we provide, within a personality neuroscience framework, a short but recent spot on the brain-personality relationships, focusing on personality-related individual differences in cortical activity during emotion regulation and social interactions. Neuroscience of personality is a new field of study within the cognitive and affective neurosciences, enclosing molecular genetics, psychopharmacology, neuroimaging, and psychophysiological methods as its tools to study the biological basis of personality and to develop explanatory models of individual differences. Researchers have begun to use a variety of available personality trait measures showing that personality modulates excitatory and inhibitory processes through connections between the most important brain nodes involved in social cognition, attention regulation, decision-making, and executive control. The present symposium provides a recent integrative view of brain-personality relationships, focusing on personality-related individual differences in cortical activity during emotional regulation and social interactions. In this perspective, Gennady Kniazev of the Scientific Research Institute of Neurosciences and Medicine, Novosibirsk, Russia, in his presentation titled Personality in Self and Other Referential Processing, Fmri Functional and Affective Connectivity Study, will report his recent research findings focusing on Big Five related individual differences in self and other referential processing using fMRI functional and affective connectivity. Joseph Ciociari of the Brain and Psychological Science Research Center, Swinburne University of Technology, Australia, in his presentation titled The Desire of Fame, an Eeg investigation of individual differences, will report his Eeg and neuroimaging data as correlates of materialism, desire for fame, and extrinsic aspirations. Almira Kustubayeva of the Center for Cognitive Neuroscience, Kazakh National University, Kazakhstan, in her presentation titled ERP Study of Decision-Making and Executive Control in Patients with Major Depressive Disorder, Individual Differences and Depression Subtypes, will present her recent ERP findings depicting individual differences in major depression subtypes in decision-making and executive control. Vilfredo De Pascalis of the Sapienza University of Rome, Italy, in his presentation, Influences of EEG Activity, Heart Rate Variability and Personality Traits on Placebo Analgesia During Self-Pain and Empathy for Pain, will report his recent correlation data finding on the influence of oscillatory EEG activity, heart rate variability, and personality traits on placebo analgesia of self-pain and empathy for pain. Thank you.
The nature of self is one of the most controversial questions throughout the history of philosophy and science. Different approaches emphasize different aspects of this concept, including emotional, cognitive, and social self. Moreover, some authors deny its reality altogether, claiming that the self is just an illusion. One of the most popular approaches to the study of self is contrasting self-referential with other referential processing in the trait-adjective judgment task. In this task, participants are asked to judge whether trait adjectives properly describe the participants themselves or some other person. Most of the studies using this paradigm investigated regional activation effects. Functional connectivity analysis brings out conceptually different kind of evidence. A search of connectivity research is associated with discovery of the so-called resting state or intrinsic connectivity networks, which are spatially separate but functionally related regions exhibiting temporal correlation even in the absence of a task. Perhaps the most intriguing theme in the study of ICNs is the contradistinction of task-positive and task-negative network. The default mode network, or DMN, has been labeled task-negative because it decreases its activity in most kinds of tasks that require attention to the environment, in contrast with most other ICNs which show activity increases in such kind of tasks. Perhaps most important of these latter networks are the central executive network and the salience network. These networks are involved in sustaining attention in the course of a task and directing it to most salient stimuli, respectively. The task-negative and task-positive networks are supposed to correlate negatively with each other, and an appropriate balance between their activities is presumably a prerequisite of mental health and adequate brain functioning. TMN is the primary network associated with internally oriented attention and self-referential processing, but it may be involved in a broader range of social and other cognitive processes as well. Central executive and salience networks are mostly associated with externally oriented attention, but parts of these networks could be involved in explicit self-referential processes. Much evidence shows that a dominance of the DMN or the TPN is observed in pathological conditions such as depression, social anxiety, and PTSD, and this is interpreted as a consequence of enhanced self-focus and diminished attention to the environment. On the other hand, meditation practices targeted at diminishing self-focus and maintaining attention to the immediate experience have been shown to decrease DMN and increase TPN activity and connectivity. It could be expected that differential involvement of the DMN and the TPN should show up in the context of self and other referential processing as well. It is important to emphasize that different others have different degrees of closeness to the self. The overlap between self and other representation has been repeatedly noted, and the degree of this overlap may be modeled by a single dimension spanning from self-centeredness on the one pole to self-other connectedness on the other. It could be hypothesized that self and close other referential processing may engage the DMN more than the TPN, whereas distant other referential processing may engage the TPN more than the DMN. A direct comparison of relative involvement of DMN and TPN in the processing of self and different others, varying in the degree of their closeness to the self, is lacking in the literature. In this study, using Fmri function connectivity data, we aim to directly compare DMN versus TPN connectivity during self and other appraisal in the trait adjective judgment task for different others varying in the degree of their closeness to the self. We hypothesized that the DMN versus TPN balance should linearly decrease during evaluation of self, close other, distant other, and an unpleasant person. 50 undergraduate and postgraduate students were initially enrolled. 3 participants were later excluded from the analysis due to excessive Fmri artifacts, thus leaving 47 participants. For the trait adjective judgment task, 150 adjectives were taken from personality questionnaires and descriptions of appearance. 35 experts rated each adjective using 5-point Likert scale on emotional valence and emotional arousal. Based on these average ratings, 30 positive, 30 neutral, and 30 negative adjectives were selected so that they did not on average differ on length and the number of vowels. Participants were asked to choose the best friend, a neutral stranger, and an unpleasant person. Next, they were presented with adjectives and asked to judge whether the respective trait applied to the evaluated person. Whole brain fMRI data were acquired with an EPI sequence on a 3 Tesla scanner. The analysis of both task-related bolt activation and task-related functional connectivity was performed using a block design. To account for the effect of negative, neutral, or positive emotional valence, we used parametric modulators along with parametric modulators coding the subject's response. After preprocessing for each subject, a general linear model was set up by specifying the onsets and durations of the 4 task conditions as boxcar functions. Single subject's hemodynamic response was modeled by convolving the boxcar functions with a canonical hemodynamic response function. An important question is whether the observed effect depends solely on the content of a task or is confounded by task difficulty. To control for task difficulty, we included reaction time as an additional parametric modulator in the design matrix of both bolt activation and connectivity analysis. Modeling the effect of the degree of target's closeness to the subject self was performed using a T contrast and attributing to the 4 targets linearly decreasing weights. Participants' age and sex were entered as second-level covariates of no interest. In the event-related analysis, presentation of each adjective was modeled by a boxcar function with on and off points corresponding to the time at which the adjective was presented and the time when the subject pressed the response button, respectively. This function was convolved with the canonical hemodynamic response function, and the GLM design matrix for each subject included separate regressors for negative, neutral, and positive adjectives within each of the 4 conditions. After model estimation, contrast images representing the effects of each adjective valence within each experimental condition were computed for each participant and submitted to a second-level random effects analysis. Task-related functional connectivity was analyzed using a block design. The 3 parametric modulators were also included in the first-level design matrix. 4 seats were selected to represent the DMN, and other 4 seats represented the TPN. Maps of Fisher transformed bivariate correlations between the seat ROI time course and all other voxels in the brain were used in the second-level GLM analysis. The factorial design included 2 within-subject factors, task and network. T-contrast was used to model a linear relationship between the degree of self-closeness and the degree of DMN dominance. Participants H and sex were entered as second-level covariates of no interest. False positive control was implemented through a combination of voxel level height threshold and cluster level extent threshold. Repeated measures ANOVA with target and adjective variance as factors and the number of choices as the outcome showed a significant interaction between the factors, which indicated that negative characteristics were more frequently selected for the description of unpleasant person and positive ones in all other cases. Friend received the highest and the unpleasant person received the lowest overall score. Modeling a linear relationship between the degree of self-closeness and brain activation yielded a significant effect in parietal cortical regions. The opposite effect was not significant. In the center of the significant cluster, activation was higher in me than in friend than in stranger, and in stranger than in unpleasant person condition. Now I will describe results of GLM analysis of bold activation using the event-related design. The main effect of adjective variance was significant in 5 clusters. In the posterior cingulate cortex, left angular, right supramarginal, and left postcentral gyri, the activation was highest for positive and lowest for negative adjectives. The opposite pattern was observed in the right precentral gyres. The interaction between target and adjective variance was significant in the left and right sensorimotor areas. Recall that activation in the left sensorimotor area reflects the preparedness to press the right hand button, which corresponds to the yes response, whereas activation in the right sensorimotor area reflects the preparedness to press the left hand button, which corresponds to the no response. As the slide shows, the subjects tend to respond yes when they are presented with positive descriptions in the me, friend, and stranger conditions, and negative descriptions in the unpleasant person condition, and to respond no in the opposite cases. To highlight the correspondence between the behavioral and bold activation results, I show them here side by side. The principal test of our hypothesis was performed by specifying a double contrast, me greater than friend, greater than stranger, and greater than unpleasant person, plus DMN greater than TPN. This contrast yielded 3 significant clusters, 2 of which were situated in posterior cortical midline regions and 1 in the left middle frontal gyres. The degree of DMN dominance was higher in me than in friend than in stranger, and in stranger than in unpleasant person condition. The theme of DMN-TPN relationship has been most thoroughly discussed in the context of psychopathological disturbances. A dominance of the DMN over the TPN has been demonstrated in depression both in clinical and non-clinical samples. This dominance correlated with the severity of self-focused rumination, which is a consequence of inherent in depression enhanced self-focus. On the other hand, engaging in a demanding activity gives rise to the experience of flow, which is accompanied by deactivation of DMN and activation of TPN regions, and a positive experience of pleasantness and intrinsic motivation. Meditation-related changes in DMN-TPN balance are accompanied by metacognitive capacities of decentering and self-transcendence, which essentially mean diminished self-focus and increased attention to the environment. This evidence allows to interpret our results in such a way that the DMN-TPN balance in the trait adjective judgment task reflects the balance between seeing the evaluated person as a part of selfhood versus an external object. To some degree, this balance may reflect the distinction between a subjective and an objective point of view, or between first and third-person perspective taking. An alternative interpretation could be that the observed effects actually reflect differences in emotional variance attributed to different targets. Indeed, all three networks are in different ways associated with emotion processing. The DMN hubs have been linked to social and affective cognition and are anatomically connected to regions involved in emotion generation. Moreover, the DMN is active when people experience complex emotions about other psychological qualities. The central executive network is involved in executive control functions, including emotion regulation, and the salience network also plays an essential role in emotional processing. Although it is difficult to disentangle the effect of target's closeness to the self and the effect of emotional variance attributed to the target, some results do not fit this interpretation. In particular, the DMN-dependent balance is higher in the me than in the friend condition, despite the fact that emotional variance attributed to the target is higher in the latter case. Besides, we performed all the analysis controlling for the effect of emotional variance. An important question is to what extent differences in brain activity and connectivity could represent more general differences in task difficulty rather than differences in self-relatedness per se. It would make sense that assessing adjectives for less familiar people would be more difficult than for themselves and close others. It has been shown, for instance, that the task difficulty might affect whether or not brain activities within MPFC would be dissociated between self and other referential. Reaction time is frequently used as a proxy for task difficulty. We therefore performed all the analysis controlling for RT. In any case, some results are difficult to explain in terms of task difficulty. Thus, mean RT was higher in me than in friend condition, implying a greater difficulty in the former than in the latter case. This seems reasonable because self-evaluation needs an ability to look at the self from a third-person perspective and should be more difficult than evaluation of a well-known close friend. Nevertheless, both bolt activation and connectivity analysis showed that the DMN dominance was higher in the former case. Moreover, it has been shown that task difficulty specifically affects MPFC activity, whereas in our study, activation differences were found in the PCC and precuneus. Some limitations of this study have to be discussed. Our experimental design somewhat differed from the majority of other studies using the trait adjective judgment task. In many studies, a control condition was included, and the person of distant other was frequently represented by a known political figure. We have not included these tasks deliberately, because we were interested in comparing conditions with each other rather than with the control. Using a known political figure to represent the distant other would be disadvantageous in our case, because the perception of such a figure is frequently associated with emotional variance ranging from love to hate, and thus muddying the distinction between close and distant other. Summing up, this is the first study that investigated the association between DMN-dependent balance and the degree of self-relatedness in the trait adjective judgment task using Fmri functional connectivity data. We hypothesized that self and close other processing would engage the DMN more than the TPN, whereas distant other processing would engage the TPN more than the DMN. A positive association between the degree of self-closeness and the degree of DMN dominance was revealed in the posterior central midline structures and in the left dorsolateral prefrontal cortex. These results show that the association between DMN-TPN balance and self versus other focus shows up not only in psychopathological conditions or in meditational practices, but is present in mainstream population in ordinary psychological processes related to self-cognition, related to social cognition. So, thank you for attention.
Welcome everyone to my talk, The Desire for Fame, an EEG Investigation of Individual Differences, as part of the Symposium of Progress on Personality Neuroscience Research and Individual Differences, chaired by Professor Vilfredo Di Pascalis. I'd like to acknowledge my two colleagues, Jeffrey Pfeiffer and John Guntis, who contributed to this work, and I'd like to thank the organisers of the IOP 2021 conference, this virtual conference, and thank you for making this all happen. Okay, so throughout history, populations have idolised individuals for a range of reasons, and the desire for fame may be a driving factor. Recent studies have investigated these. Social media, television, and the rising popularity of reality TV shows and the use of some celebrity endorsements impact on consumers' decision making. The major motivators in the desire for fame seem to be lifestyle aspirations, financial success, others' approval, social recognition, and the ensuring self-acceptance and the impact of celebrity. Been a number of studies which have demonstrated that quite clearly. But what about from a cognitive neuroscience point of view? What does this mean in terms of individual differences? It seems that individuals higher on the desire for fame may demonstrate a higher increased attention, memory access, and reflection regarding the image displayed. Frontal and temporal regions have previously been associated with famous face processing and thought to relate to accessing semantic memories. And areas of the brain associated with processing aversive risk may also be active during the decision phase. And it seems that maybe the desire for fame may be reflected in the decision-making processes associated with attraction to fame scenarios or reward mechanisms. So the current EEG study aimed to investigate the individual differences of people who are more likely to have higher scores for the desire for fame and extrinsic and intrinsic aspirations. This study focused on the personality facets associated with the desire for fame survey instrument, first published in Guntis et al. 2012, by examining the electroencephalogram during decision making associated with a specifically designed desire cognitive task. This examination of the confluence of a social psychological personality paradigm, or the intrinsic-extrinsic motivation, with a psychophysiological element, or the EEG data, was done to provide insight into some innovative possibilities for consumer neuroscience and social psychology. The protocol that we used consisted of three components. That is, we looked at ERPs and alpha and beta coherence analysis, similar to some of our previous work. We also looked at S-Loretta, beta in particular, based on the work by Cook et al. 2011, and we used a desire cognitive task, specifically designed paradigm. So the number of participants, 23 participants, 11 male and 12 female, between the ages of 22 and 35, were taken from a general university community sample, and they were already on an existing database for the Desire for Fame survey. They had no neurological disorders, and the research has been ethically approved by the University Human Ethics Committee. We used two psychometric scales, as mentioned already, the Desire for Fame, and we also used a personality orientation scale as well. And this particular scale looks at four thinking styles or thinking orientations, which I'll talk about a little bit later. And the way in which we looked at the Desire for Fame, once we had everyone scored, the median was 2.3. Those people who were higher than 4 were in the high group, and those people who were lower than 1.4 were in the low group. The tasks was a specifically designed cognitive task, which had famous and non-famous visual imagery, and it came from a particular database. Epoch average for yes and no responses were taken for would you like to be this person, yes or no. So the desire for fame is this particular scale here, and it looks at financial success, attractive appearance and social recognition, looks at self-acceptance and affiliation as well, and in terms of the general desire for fame, it's anticipated outcomes associated with status, power and happiness with an alpha, Cronbach alpha of 0.86. The personality orientations I mentioned earlier are these. Those who fall into a more thinking, logical style of processing, material or physical personality, more emotional feeling in terms of their thinking styles, or those who are more imaginative, intuitive, and they're the Cronbach alphas for each one. So just in terms of EEG recording, very standard 32-channel EEG recording using Neuroscience system 4.3, mastoid nose reference and forehead ground electrodes. It's just a common average reference. And we also recorded EOG because there's a fair bit of eye movement associated with the task itself, so we wanted to make sure that we could remove any artifact during offline analysis, and we used ICA to do that. The gain was 100,000, and we filtered the data from 0.1 to 50 Hertz. And the speed of acquisition, 1,000 Hertz. So we did a fair bit of offline analysis for artifact, EOG, and we looked at various epochs, decision-making epochs, and we looked at the associated VEPs or the ERPs, and we also calculated EEG band activity. So during the task itself, we had epochs of yes and no responses for high and low desire for fame, and we used a 2-second window before the response as a baseline. The EEG analysis, we looked at specific ERPs associated with these particular electrode sites, and we looked at N1, P1 and P3 for both amplitude and latency. We looked at alpha band EEG for the coherence analysis, and we looked at beta activity with respect to the S-Loretta source localization. The stats we used were multiple regression for personality orientation, aspirations and goals, and a variety of ANOVAs and t-tests we used to look at the EEG. So the presentation paradigm was more of a block design. So people would look at a screen, fixate on a spot, and they were given certain instructions. There's some text describing the next picture for about 5 seconds. The picture would be presented, and the person then had to answer yes or no, would you like to be this person? There were over 180 trials associated with each of the conditions, and the 3 conditions were images that are non-famous and inappropriate, famous without achievement, and then famous with achievement. So there are 3 different categories of famous aspects. And the inappropriate are associated with the images of people taking chances, risk-taking behaviours. So here's a breakdown in terms of the task timing sequence. So the image would appear and the ERP would be calculated for 600 milliseconds, and the coherence epoch was the full 4,000 milliseconds after that. So as mentioned already, that we used that 600 milliseconds to calculate the visual ERPs, and the 4 seconds was used to calculate the coherence data. And people will ask the question yes or no. And we had a 2-second interstimulus period. When we have a look at the results, that's just the behavioral results just to begin with, we find the following, that we have our 4 categories of personality thinking styles, the physical, material, thinking, imaginative and emotional thinking styles, that we did a neuroimaging study associated with that and published that in 2019. And we were able to distinguish the areas of activation predominantly associated with these thinking styles, and we found that the physical and material, or the person who's more pragmatic or materialistic, for example, tended to, in this case, we found a correlation between the physical material orientation with all these variables associated with the dependent variables associated with the desire for fame. And you can see the materialism, desire for fame, financial success, attractive appearance, social recognition seem to be very important for those. But notice as you go across the different personalities, we find that there are some correlations which are quite significant in the thinking style, the imaginative style. But interesting, those who think with their emotions first didn't tend to, there were no correlations between the personality orientation of emotion and the desire for fame, which was interesting. It's like they're a better balance, perhaps. We'll talk about that a little bit down the track. So we made these comparisons with the different fame groups for yes and no response, and we found the following. We did some one-way ANOVAs and found some significant responses with respect to the no response for the P1 and P3 when we are comparing the low and high groups, quite significant. And with the yes response, we tended to find the P3 amplitude quite significant at PZ. So we contrasted the groups and we found that it seems that the N1 response was significantly early compared to those of the high desire for fame. So the high desire for fame N1 response is late in comparison. And this is very similar to what Tops had recorded in 2011. It's very consistent findings associated with that group. S-letter was then calculated during this period, and this is what we found. So for the high desire for fame, we tended to find the Brodmann area 10, which is associated with risk and decision-making, reward-seeking behaviours, was quite high. The activity, the source was there with these particular individuals, this high desire for fame group, compared to the low desire for fame group. The activity wasn't as strong, but nonetheless the cuneus, the visual processing systems, this is the back, were quite active for both. So even though that was pretty much the area that for the low desire for fame, the B17 seemed to be quite consistent. So we also did coherence analysis, and we used a particular montage that you see here. And we looked at no and yes and no responses across the group, and then we contrasted the high desire and low desire for fame groups for yes responses, and we found that this is where we had the higher differences between in alpha coherence with respect to the high and low desire for fame. Makes sense. We see this sort of activity with this supports the S-Loretta data that we found earlier, with the frontal responses being so crucially important for the high desire for fame. So that's what we found very quickly. Then if we break it down into, for the low desire for fame group, if we look at the not famous images compared to the famous but no achievement and the famous with achievement, you can see that the low desire for fame did not produce that frontal response that we saw in the high desire for fame. As a matter of fact, 3 areas came up: the Brodmann area 17, most of the occipital lobe, and the cuneus were quite active. So these people were processing the visual information, mediated perhaps by attention, working memory, and some sort of reward expectation. However, may be associated with a better inhibitory control in comparison to the high desire for fame that we see here. So you can see the frontal areas, Brodmann area 10 quite active, but also the inferior frontal gyrus. And this is generally associated with strategic processing, memory retrieval, higher executive function, but it's also implicated in risk aversion and perhaps some compulsivity associated with this particular response. So for all 3 categories of, even though we're looking at not famous imagery, famous, no achievement, famous with achievement, we tended to find this very strong frontal activation taking place, consistent with what we see with reward-seeking behaviours, compulsive behaviours as well. So what does this all mean? Well, it is only really a pilot study, and it's designed to get us thinking about where to go from here. But we see that materialism and desire for fame were found to be significantly positively correlated with intrinsic aspirations, materialism, and interest in being famous for something. And the EEG demonstrated an increased EEG coherence between frontal and frontotemporal central regions, and right frontal activation in particular, or via the S-Loretta, and this was associated with a higher desire for fame and suggestive of a very strong behavioural or compulsive response based on previous literature. So just to go through this, we find that that for all three different categories of imagery that people were shown, those associated with risk behaviours, those associated with good behaviours, and those associated with celebrities and influencers, for the low desire for fame group, there doesn't seem to be much happening except for strong visual processing as they're looking at the images and so forth. And these are mediated by visual information processing networks and mediated by attention, working memory, and reward expectation. We know that. However, when things are overly active in the cuneus, we see this in pathological gamblers in particular, and this is something we saw in the high desire for fame. For all of the high desire for fame, we did see the high visual processing networks together with the Brodmann area 10 and right inferior frontal gyrus. So we know that these areas are associated with strategic processes in memory retrieval and executive function. However, they are also very much involved in integration of information associated with sense of self and environment. So this right inferior frontal gyrus may also be implicated with respect to risk aversion, gambling, reward-seeking behaviours, or abnormal reward-seeking behaviours. So by looking at images of would you like to be famous, would you like to be like this person, those tasks, that specially designed task, we're able to elucidate these particular types of activities, this associated sense of risk-taking behaviour with just the imagery. And perhaps we may be looking at some sort of poor decision-making system in those who score high desire for fame. But however, the high desire for fame group are more open perhaps to the idea of taking risks and are prepared to be famous by any means. And this seems to be reflected in the EEG processing. However, a larger study is necessary to look at the associations with personality, and perhaps a future study would also look at the NEO-PI, looking at neuroticism and conscientiousness and so forth. So thank you for your attention. I hope you enjoyed that. If you have any questions, please ask the questions at the end during the live Q&A. If not, you can contact me on my email. So thank you very much. And the next talk is coming up right now.
Hi, my name is Almira Kusubaeva. I'm head of the Department of Biophysics and Neuroscience at Kazakh National University, and I'm going to tell about the results of our ERP study of decision-making and executive control in patients with MDD, individual differences, and depressive subtype. Depression is associated with abnormal decision-making and impaired executive control that also may be modulated by personality. Attentional deficits in depression may be associated with impaired neural networks involved with alerting, orienting, and executive control. Abnormalities in the corresponding network brain activity detected using event-related potentials may have value in predicting depressive state. Impairments in decision-making in depressive episodes impact everyday functioning. Decision-making deficits may relate to emotional dysregulation, hypersensitivity to reward and anhedonia, or hypersensitivity to negative information and depressed subjects. Depressive symptoms are associated with abnormal decision-making processes, which may be consequences of developed negative bias or anhedonia and altered brain activation. The aim of the present research was to study brain activity during performance of decision-making tasks and attentional network tests in patients with major depressive disorder and healthy participants. We investigated the relationship between subtype-specific symptoms, individual difference, and brain activation. It has been hypothesized that MDD participants will have altered brain activation to stimuli and show deficit in executive control and decision-making. Individual difference influence on brain activation and depressive score, and depression subtype also may relate to individual differences and depressive score. We had two separate studies with ANT task and with decision-making task. In the first study, ANT study, we had 93 participants divided in three groups: depressed group, risk groups, and healthy group. In the second study, decision-making study, we had 120 participants divided to two groups: a depressed group and healthy group. Here you can see from the table demographic data and the data of the severity of depression. We used psychometric methods. The next psychometric methods: inventory of depressive symptomatology, trait meta-mood scale, positive negative affects schedule, the Dundee stress state questionnaire, emotion regulation questionnaire, and rational experiential inventory. The ANT task was programmed in E' software. Original ANT was modified to increase the number of trials to 96 in 9 periods, 864 in total. A modified version of ANT task was used in the previous study and adapted to local population. Furthermore, feedback was added after each trial. Adding feedback extended the duration of the experiment to 70 minutes. The search and rescue tactical decision-making task was programmed in E' software as well, and it was adapted to the EDRP experiment. The participants performed a rescue operation with the goal to find as fast as possible a lost party of researchers in the Antarctic and save their lives. The participant was required to make 30 successive road choices trials to find the fastest road while driving a snow-cut vehicle to the location of the lost party. Each trial presented a map of the terrain with the 2 alternative color-coded roads and 4 icons with potential hazards and benefits on the roads, a statement about probability of loss and gain and their percentage likelihood in each road appears to the participant when they place a cursor over the icon using the mouse. Participants, after accessing the information of probabilities of cost and benefits, were prompted to make a road choice by clicking on 1 or 2 color-coded circles presented on the display. Information about the results of choice was given after each road selection following each trial, which contained an image of occurred event, a statement of time they had been lost or gained. The feedback screen described overall time lost and gained across all trials, with an image of easy contented and discontented explorer. EG markers were added to E' task on certain categories of the task stimuli: the start, benefits, hazards, choice making, and feedback. Here you can see the details of EG acquisition and preprocessing. We used recording from the 16 electrodes by using neuron spectrum for system, and artifact-free epochs were analyzed for the decision-making task with 200 millisecond pre-stimulus and 400 millisecond post-stimulus intervals, and for the executive control task with minus 700 millisecond pre-stimulus and 700 millisecond post-stimulus. And we analyze amplitude of P300, N100, and P100. This slide shows psychometric results in ANT experiment. As you can see here from the figures, psychometric data like energetic arousal, hedonic tone are higher in the healthy group, and opposite tense arousal and anger frustration are higher in MDD group. Trait and trait anxiety also showed significant differences. Positive and negative scales before and after experiment as well were significantly different between three groups. Here you can see the individual difference between three groups in the emotion regulation. The depressed group more often use suppression as emotion regulation strategy. Also, you can see the difference in extroversion, with the highest score in healthy group and lowest score in depressed group, and in neuroticism with the high score in depressed group. This graph shows us the significant correlations between the IDS score and state scores. In the red, you can see negative correlations, and IDS score negatively correlated with energetic arousal and hedonic tone, positive affect before task and after task, and positively correlated with the tense arousal, anger frustration, and negative affect. This graph shows us significant correlation between IDS score and trait scores. You can see the positive correlations, depressive symptom with the neuroticism and trait anxiety, and negative correlations with extroversion, agreeableness, consciousness, openness to experience, rational ability, and rational engagement. Behavioral data didn't show significant difference between three groups in alertness orienting in executive control. However, there were several significance in executive control during the 9 periods. It means the higher executive control score were observed the 3rd and 8th periods, which means worse executive control. Here you can see significant difference in P300 mean amplitude on congruent and incongruent targets in 3 groups. Higher amplitude for both congruent and incongruent stimuli we observed in healthy group. In red line, you can see red line, and low activation in the risky and depression group. You also can see 2D map of mean P300 amplitude for congruent and incongruent stimuli. Both shows us the same tendency. The same tendency were observed for the P300 amplitude no-Q and double-Q conditions in 3 groups, higher in healthy group and significantly lower in depressed and risky group. There were significant correlations between neuroticism score and ERP amplitudes. You can find results on these figures. Neuroticism were negatively correlated with executive control on CZ and executive control on FCZ, and with executive control on CZ on P300. The positive correlations, significant correlation, were found between agreeableness, openness scores and 100 amplitudes. Next, I would like to introduce the results from the decision-making experiment. As you can see, psychometric data repeated the same tendency as an ENT experiment. We found significant differences in the SSQ scales between MDD and healthy subjects, in trait and state anxiety, and positive and negative scales before and after experiment. As well, there are some significant difference in neuroticism and suppression, emotion regulations, and also attention to emotions. Similar to ANT experiment, IDS score showed positive and negative significant correlations with the DSSQ scales, and negative and positive affect state anxiety before and after experiment. Not so many significant differences were found in behavioral results. As you can see from the figure 13, there is a significant difference between MDD and healthy group in reaction time to benefits, higher in MDD group. Results in ERP data were similar to ANT study as well. P300 amplitude in MDD group were significantly lower compared to healthy group. You can see on the figure ERP at CPZ, and there is significant difference in P300 amplitude between two groups. On the left side, to the map of the mean P300 amplitude, it's clearly seen higher activation in the healthy group. P300 amplitude on feedback also significantly correlated, negatively correlated to neuroticism. Regression analysis with dependent variable P300 amplitude in CPZ and the predictors neuroticism and depressive score showed significance. The next bar show us the relations neuroticism with P100 amplitude on hazards as well. Those relations opposite to the relations to P300 amplitude on feedback. It may be because of a specific bias to the hazard. Also, little attention has been paid to the fact that recognized depression subtypes such as melancholic and atypical depression differ regarding mood reactivity to emotional stimuli, and whether this is related to underlying neuronal difference between the subgroups of depressed patients. Therefore, it is important to examine the neurophysiological difference related to the subtype, and we divided our depressed group to the two subgroups with the highest score in melancholic subtype and highest score in the typical subtype. And we found significant differences in the ERP in P3 in the ERP in P100 amplitude on hazard. You can see from the figure 18 that the reaction to the hazard were higher in the melancholic group compared to the atypical group. And as well, there is a significant differences between the positive and negative conditions in two groups. Another interesting finding was that the melancholic score negatively related with extroversion score, and openness negatively related to the P100 amplitude on hazards. Our conclusions. The ANT study confirms that depression is linked to deficits in brain networks for alerting and executive control. Decision-making studies suggest basic attentional deficits are modulated by emotional factors. Depression attenuates P300 response to positive but not negative feedback. Depression effects on earpiece may in part reflect higher neuroticism. Both studies showed neuroticism was associated with reduced P300 amplitude, and neuroticism also correlated with larger P100 amplitude to hazards. And another conclusion, the depression subtypes also influences response, stronger for melancholic subtype. The future work may reveal multiple cognitive affective deficits in depression. I would like to express acknowledgements to Ministry of Education and Science of Kazakhstan for provided grants. Thank you for your attention.
Influences of EEG activity, heart rate variability, and personality traits on placebo analgesia during self-pain and empathy for pain. Aims. We wanted to extend our previous placebo analgesia findings observed for tonic pain to phasic pain responses. More specifically, using principal component analysis, we wanted to detect the significant physiological factors, including heart rate variability indexes and non-phase locked event-related EEG oscillation that can significantly explain a substantial proportion of the variance in phasic self-pain reduction and empathy for pain reduction. A further aim was to highlight significant association, if any, of motivational personality traits and empathy traits with phasic self-pain and empathy for pain induced by placebo analgesia treatment. Then, using mediation analysis, we wanted to evaluate the influence of the above-mentioned physiological factors on self and other pain reduction scores, considering the mediation effect of the fight-flight freezing system and empathy traits in this relationship. 63 right-handed volunteers, 31 men, were enrolled in the present study. The study consisted of two sessions. In the first session, we administered the reinforcement sensitivity theory personality questionnaire, providing a measure of the behavioral approach system and its component, a measure for the behavioral inhibition system, and a measure for the fight-flight freezing system. The empathy component questionnaire was also administered that provide a measure of cognitive empathy, affective empathy, empathy drive, and total empathy ability. The state anxiety inventory was also administered as a control. In the second session, participants received two treatments, pain control and placebo analgesia and double-blind. Placebo analgesia included a pain manipulation procedure that consisted in the administration of a fake analgesic pill presented as a potent analgesic to reduce the pain. Each treatment included both self-pain and empathy pain condition. Treatments and condition were all counterbalanced. The experimental trial that we asked to fulfill to each participant is presented here and consistent in the presentation on the computer screen of an arrow indicating the target of the upcoming electric stimulation, to the self or to the other, the confederate, that was also present during the stimulation. This arrow was followed by an anticipation cue indicating the intensity of the upcoming electric stimulus. Yellow spark upcoming painful, blue spark upcoming non-painful. After 3,500 milliseconds, a delivery cue was presented for 1,000 milliseconds. Red spark represent painful stimulus. Green spark represent non-painful stimulus. After the deliberation of 6 pulses, a train of 6 pulses that was painful and non-painful, the participant was required to rate the level of experienced pain and the level of experienced distress in a scale from 1 to 7 points, and this was done for the self-pain and for the other pain. During this task, we record the electrocardiogram and the electroencephalogram. And after, we analyze the heart rate of the participants through heart rate variability processing that provide the mean interbeat interval, the standard deviation of interbeat interval, the low frequency peak and the high frequency peak of the Fourier transform of the interbeat interval, and also the sampling entropy. The G was recorded in the conventional way, except that we, for each epoch, selected epoch, artifact-free epoch, we subtracted the average ERP for that condition to avoid epoch responses in our analysis, because we use non-phase locked event-related oscillations to study pain and other pain effect on EEG. Continuous wavelet transform were used, using Morlet complex instantaneous amplitude and with Gabor normalization. With this transform, we set the value of Morlet parameter C equal to 5, and we calculate normed power of event-related oscillation, and we also provide an induced normalized of non-phase locked time frequency power representations for each trial, for each epoch. And after this, we use the peak of the extracted frequency of interest for further analysis. Results we obtained in terms of ratings, we found reduced pain rating for the self condition and reduced distress rating also for the self condition, and while for the other for the empathy condition we didn't find significant effect for pain, but we did find for distress rating, and here is presented. We also found for self-pain a gender effect. The female had more pain than the male. In terms of time frequency representation, we obtain this configuration for pain and placebo condition in the self condition and the other condition, the empathy condition. And we did the difference between these representations, and we found that the main frequencies in this difference were the 7 Hz, 12 Hz, 18 Hz, 27 Hz, and 40 Hz. We select this frequency, and we use the extraction through the single frequency extraction from this wavelet representation for further analysis. This extraction is non-phase locked power, and is represented here, and we see how it's changed across frequency, the amplitude, and across also pain and placebo condition. In order to select the locations across the scalp, we calculate the current source density in order to have a representation of the location of the generators, and in the end we obtained this representation in different time interval, and this representation were more often located in the midline, and thus we select the FZ, CZ, and PZ as electrodes to study this. For each frequency of interest, a natural logarithm transformation was applied. We performed separate principal component analysis on pain minus placebo difference scores for heart rate variability indexes, interbeat interval, standard deviation of interbeat interval, low frequency peak, high frequency peak power, and sampling entropy, and also for power peak values for 7 Hz, 12 Hz, 18 Hz, 27 Hz, and 40 Hz at FZ, CZ, PZ during self-pain and other pain. We obtained a number of physiological factors that were correlated by accounting for gender and anxiety, were correlated with the behavioral approach system measure, with the empathy trait measures, with pain reduction and distress reduction in self-pain and other pain conditions. Among these significant correlations, we found in some physiological factors that were here reported in red, that were correlated with behavioral inhibition and pain reduction and distress reduction in the self-pain condition. And also we found the 27 Hz midline power correlated with total empathy ability and with numerical pain differences scores. Since these factors reported in red were all intercorrelated, we had to select the ones that was most reliable because we had a problem of collinearity. We test the collinearity and we found the significant effect for collinearity. So we applied the elastic net methods to solve this problem, and the two factors that were highlighted by this analysis were the time heart rate variability and midline 7 Hz power factor and the midline 27 Hz power factors. These two factors then, for the 7 Hz power difference scores and 27 power difference scores, we applied a Loretta transform to evidence the most involved area in the self-pain condition and in the other pain condition. In the self-pain condition, we found involved the BA40 here. That was mainly in the left hemisphere because the stimulus was in the right. This in the self-pain condition. In the other pain condition, we found the anterior cingulate cortex more involved, the BA32. This is an interesting finding, since in the next we found a significant relationship between the fight-flight-freeze system and pain reduction in the self-condition, the self-pain condition, and a significant relationship, but positive relationship, between total empathy ability and pain reduction in the other pain condition. These were interesting findings that we used for further analysis, further mediation analysis considering the physiological factors of time, heart rate variability, and midline 7 Hz power that was a good predictor of self-pain reduction. And this effect was not only direct, but also direct. The indirect effect was involving the behavioral inhibition system that in turn influenced the fight-flight-freeze system and then influenced pain reduction. So we had two effects, one direct, another one indirect, and both were linked to self-pain reduction. Gender was also a significant covariate, influencing the fight-flight-freezing system and indicated that the women were more fight-flight-freezing than the men. Next slide, here we represent the simple mediation analysis, including the total empathy ability and the midline 27 Hz power that influenced directly other pain changes in following the treatment of placebo analgesia. And we found a direct influence and an indirect simple influence through the total empathy ability. The influence of gender was that the gender they had, the women, the female, they had higher level of total empathy. They were more empathic than the male. This is all. So, in conclusion, taken together, these findings suggest that empathy for pain reduction rely on neutral and autonomic processes that are in part functionally different from those engaged by the first-hand experience of pain and involve different personality traits. Future studies should examine the potential role that these individual differences measures may play in patient responsiveness to acute pain. Thank you.